Hello students, in the last class we have discussed about carbohydrate. Now we will discuss about the protein. What is here protein? Protein is actually the polymer of amino acids. Protein is the polymer of amino acids. And first, so to know about the protein, we will have to firstly be acquainted about the amino acids. Firstly, what is amino acids? So amino acids are the molecules in which the amino group and the carboxylic acid group both will be bonded to the same carbon atom. That is called amino acid. So what here structure? So there will be a carbon atom. In amino acids, there will be a carbon atom in which there will be an amino group, that is NH2 group. NH2 group and NH2 group, here carbon and carboxylic acid group. So remaining here alkyl group. This one and one hydrogen. It is amino acid. Okay guys, that is amino acid. That is the compound in which the amino group and the carboxylic acid group both are bonded to the same carbon atom. That is amino acid. Now amino acid here about the structure, and that is more important for the next importance here. The more important for your CUT ET examination and the NEET examination. Here R means any group bonded to the carbon atom. Here if hydrogen will be there, the simplest amino acid is glycine. That is important for you. Okay, nowadays it has been asked. No, not in 12th. That means I think have not asked in the 12th board examination. But that question was asked in CUET examination. In 12th board examination, maybe in the upcoming time may ask, but has not been asked till now, according to my knowledge. As I have seen the question banks, till now it has been not asked in the 12th board examination. But in the 12th board examination, in the CUT examination, it was asked, and in NEET examination it was also asked. So simplest amino acid is glycine. In the glycine, on the place of R, just let's see this amino acid here. This is that represented by this amino group and the carboxylic acid group. Both are bonded to the same carbon atom. One hydrogen here and here alkyl group. On the place of alkyl group, if there will be hydrogen, then that will be called glycine. So here that will be this one. Here hydrogen. So it will be called glycine. Till now 20 types of amino acids has been discovered till now, naturally occurring, that is. So of the glycine is the simplest amino acid in which the hydrogen is placed on the place of R, that is glycine. If there will be methyl group, then that will be called alanine. So on the place of here hydrogen, if there will be CH3, then that will be called alanine. So that will be not a glycine, it will be not a glycine if on the place of hydrogen, then glycine. If on the place of alkyl group, that means R, on the place of R, if there will be methyl group, then that will be called alanine. Same, if there will be isopropyl group on the place of this R here, if there will be isopropyl group on the place of R, then that will be called valine. So simply essential amino acid is valine. Okay, I tell you essential and non-essential. Okay, again. That will be called valine. Okay, guys, and like that, you could solve here go the chart, and particularly those students who are preparing for the NEET examination, the CUET examination, as well as for the IIT examination, must practice this one by adding these groups here given already on the place of R. Okay, so most essential amino acid is glycine, and most that is simplest essential acid is glycine, and the simplest essential amino acid is valine. The simplest amino acid is glycine, and the simplest essential amino acid is valine. Okay, first of all, what is essential amino acids? The amino acids which do not synthesize in the human body and must be taken in the diet. So the amino acids which do not synthesize in the human body, but amino acids is too much important for our body because that is the monomer of protein. So amino acids here, the amino acids which do not synthesize in the human body, so it will be essential to us to consume that, the source in which those amino acids occurs. So that is called essential amino acids. So essential amino acids are the amino acids which do not synthesize in the human body and hence must be taken in the diet. And non-essential amino acids, what is that? So non-essential amino acids are the amino acids which could be synthesized in the human body. Okay, so alanine can be synthesized in the human body. That is here. Then after this here, and here, as what are the essential amino acids? The students who are preparing for the NEET examination, particularly that students, must write that here. This one here, star marked is of the essential amino acids, non-star marked is non-essential amino acids. So make a diagram. That's I leave the work on you. Some work you also do. That is here, make a here chart of the essential amino acids. Okay, and revise them again and again, and I will not tell that means 5 times or 10 times here till you all the amino acids will not you mucked up. Till then you revise them. Okay, and suggest you that will be more effective, that if you have practiced suppose that in the morning today, then in the evening before taking the sleep also write that, and in the next morning and in the night, and then after you may leave for the 4-5 days, but after 4-5 days again watch out, and during that means in a regular interval of time period you watch out them again and again. Okay, so then the whole information will remain in your memory. So now let's see, and that memory tips will be not only for this one here, that will be all the information which you will have to only mugged up, and you cannot do anything except the mugging them. Isoleucine, arginine, this one is here, so make a list of that, essential amino acids and non-essential amino acids, no need to do that. Okay, in the examination question, in the 12th board examination, question has been asked, that is, what is essential amino acids? I have told you already, the amino acids which do not synthesize in the human body and hence must be taken in the diet. Okay, that is called essential amino acids, and give their example. If we'll ask non-essential amino acids, so the amino acids which synthesize in the human body and hence if will not consumed by the human body, then it will not get affect a lot to the human being. Okay, that is non-essential amino acids, and you give the example. Okay, if you will provide the example, then you will get full marks. Now let's see. Then after it is about the protein. Protein is what here? Okay, this is here reaction bitter ion Hodge forms. So obviously in this case here, what happened? How the bitter ion forms? So when hydrogen of this carboxylic acid that goes toward this NH2, and that become NH3 plus here and O minus here. So there is formation of bitter ion. So that is okay. Now let's see about the structure of protein. Okay, protein, about the protein, I reiterated that protein is actually the polymer of amino acids. Polymerization takes place. So let's see. Suppose alanine, I'm making this one short. And making a polymer, that means dipeptide, in which that will be of glycine and alanine. So listen here. For the glycine, hydrogen will be there, and alanine on the place of hydrogen there will be methyl group. You already aware. So here also I am taking of alanine. Any other protein like that here? Okay, not necessary there will be glycine and alanine only. What happened? Hydrogen of this amino acid group that combined with OH form H2O. In the same way, hydrogen of this amino group that will combine with this OH form H2O. Again, hydrogen of this amino acids will combine with other COH group of the other amino acid group, and same here, this hydrogen will combine. Oh, sorry, this OH will go. So what happened here? So during that course, how the structure will be? C double bond O here, this one. From here it will continue, and from here it will continue. So this bonding, that is called peptide bonding. The question of in the NEET examination was asked that in protein the amino groups are bonded with each other through which type of bonding? So answer will be peptide bonding. Okay, options I have forgotten, and you don't need what was the option there. So simply here that in the protein the amino groups are bonded, amino acid groups are bonded with each other through peptide bond. So this is peptide bond. Peptide bond, it is just like this peptide bond is just like that of the amide. How is amide? C double bond O is bonded to NH. That means nitrogen is doing bond formation with SLE carbon atom. So that is amide. So that is the structure of here. In this case, there will be formation of peptide bonding. There will be dipeptide if only two amino acids will combine together. In the same way, tripeptide if three amino acids will combine together. That means that will be called simply peptide there and after 10, usually I'm telling, it's not necessary, usually 10. After that, a number of amino acids combine there, and that will be called after the 10 amino acids, that will be called polypeptide. Okay, that will be called after 10 when when the amino acids greater than 10 in number will combine together. When the amino acids greater than 10 in number combine together, then the compound form that will be called polypeptide. Okay. Then after this, what is here dipeptide, polypeptide, that was, and there is here, but it's not necessary that if greater be the number of amino acids, then that only will be called protein. Usually that happens when the number of amino acids will be more than 100 in the chain, polypeptide chain, then after that is called protein, usually. But not necessary. There are a number of proteins in which the number of amino acids are less than 100. There are few proteins in which the number of amino acids are less than 100. As for example is insulin. Insulin here, and I hope all of you know the role of insulin, because nowadays India has become the capital of diabetic. So insulin, actually I am telling them after insulin, that plays a major role for the conversion of glucose into glycogen. So insulin converts the glucose into glycogen. Okay, and that is made up of 51 amino acids. Okay, now classification of amino acids. Amino acids is of two types. One is fibrous, protein is of two types. One is fibrous proteins, another one is globular protein. Fibrous protein and globular protein. So what is fibrous protein? In fibrous protein, the polypeptide chain run parallel to each other. They run parallel to each other, and each chain are bonded with each adjacent chain are bonded with each other through hydrogen bonding or disulfide bonding. In fibrous protein, the polypeptide chain run parallel to each other and they are bonded. Adjacent chain are bonded with each other through hydrogen bonding or the disulfide bonding. Okay, this type of protein are insoluble in water because already hydrogen bonding took place here. So this type of protein are insoluble in water. Example of fibrous proteins are hairs. And so have not you realized your hairs never solubilize in water till from childhood to here, whatever be your age. So that is a type of fibrous protein. The nails is not fibrous protein. Actually that is okay, fibrous protein it is yes, but that is called keratin. Here the hairs is made up of keratin protein. And in muscles there is keratin protein. Here nails also, horns, that means horns of the animals, buffalo, cows and other goats and so on. That is horns. Hooves, they are type of keratin protein, which is a type of, that means horns, hooves, nails. These are types of keratin protein, which is type of fibrous protein. Another example of fibrous protein is myosin. Myosin. This myosin protein exists in the muscles of the animals usually. So, who is non-vegetarian? You may think that pieces there is. So, fibrous pieces you have seen in the who have taken the non-veg. So that is actually myosin protein there is, and that myosin protein there. So that do not dissolve when you cook them. Okay. So that is about the fibrous protein. Next one is globular protein. So globular protein here, three structures. That means that has been classified into three portions. One is primary structure, then after secondary structure, then tertiary structure, and then after is quaternary structure. Globular protein. So primary structure of protein here, what happened? Simply there will be formation of polypeptide chain. Okay. In globular protein, the primary structure will be the formation of polypeptide chain, and how the amino acids will be bonded with each other. In case of polypeptide chain, that is peptide bonding. So in primary structure there will be formation of polypeptide chain. In secondary structure there will be coiling of the chain. In secondary structure there will be coiling of the chain. And the second one is also in case of beta pleated structure, these coils here, this run, this polypeptide chain, this polypeptide chain, and this polypeptide chain. This polypeptide chain will will run parallel to each other, and they will be bonded with each other through hydrogen bonding. And there will be formation of ring here. Watch out this one here carefully. Watch out here, this ring, like that. So this will be like that. Here R is alkyl group. That means this R is towards right. Here R towards right hand side. But in the next ring, the R will be towards left hand side. Keep in the mind that the R in the adjacent rings will arrange in alternate manner, in right hand side or in left hand side. So in beta plated protein, the polypeptide chain will run parallel to each other here, and they will be bonded with each other through hydrogen bonding. Okay, while in case of globular protein there will be coiling of the chain. Okay. Then after, let's see about the tertiary structure. In tertiary structure, nothing else. In the tertiary structure, the supercoiling of that chain will take place. In case of tertiary structure, the supercoiling of the chain will take place. Okay. In quaternary structure, the supercoiled structure will arrange together, and each supercoiled structure will be bonded with each other through van der Waals force of attraction. So, who have taken the mutton, I am just telling, who have not taken, who has taken the mutton, so he may realize that in the fibrous protein, that means there is in the meat, some portions runs like this. And removing this fiber from this fiber will be easier. But taking out this one, that will be somehow, that means not so much easy. Okay. So here, separating this one, very easily you can separate. But to take this one, you will have to bite. By removing, but you will have to exert more force. Okay. Maybe like that here, I am telling. In the mid here, suppose this like this one. So if you will have to take out this portion, so you will have to bite, and very easily you can separate this portion, this portion, this portion, this portion. Okay, that was about the quaternary structure of protein. Okay. Now then after is most frequently asked in the examination of 12th board is about writing of the denaturation of protein. Denaturation of protein. What is that? What happened? Let's see. You know already protein has coiled structure. Protein has coiled structure in this coiling here. each pitch are bonded with each other through hydrogen bonding here. In protein, case of protein, that is these portions are bonded with each other through hydrogen bonding. When heat energy will supply or pH will change, when heat energy will supply or pH will change, then this hydrogen bonding will dissociate. As a result, the decoiling of the protein will take place. The decoiling of protein will take place, and that's why this phenomena is called denaturation of protein. Okay, what happened here? Example of this denaturation of protein is solidification of eggs. Eggs, when you might have seen the inner portion of the eggs, that is yolks and so on. They exist in the jelly form. When you apply the heat energy, then they get solidified. So that is example of that is due to the denaturation of protein. In the same way, if you leave the milk for a long period, or if you add the curd in the milk, then here what happens? That becomes thick. Your milk becomes thick and converts into curd. If you have added curd, which you can consume, and if you have not taken curd, then also something jelly form structure will form, but that you should not consume because that will create the harmful effect on your health. So that is curdification of milk. When you have added curd, then what happened? The lactobacillus bacteria will get anaerobic respiration there, and there will be formation of some amount of ethyl alcohol. In curd also there is ethyl alcohol. So there will be formation of ethyl alcohol. You know, ethyl alcohol is acidic, so pH of the milk will change, and that comes into the viscous. So viscosity of that milk will increase. But if you have not added curd and that curdification took place naturally, then what happens? The microbes present in the atmosphere that acted there and changed their pH, and hence, okay, that is not fit for you to consume for you, and that is here example of denaturation of protein. Okay, so in the examination there is that question is asked, what is write about denaturation protein? About that, what you write? Listen. Okay, I am dictating, you write. Denaturation protein. When the heat energy will apply or pH will change, then the hydrogen bonding in the supercoiled structure as well as coiled structure of the protein will dissociate, and hence this phenomena is called denaturation of protein. Example is solidification of egg and curdification of milk. Okay, guys, I tell you, have you felt or not? If you consume lots of curd, then you will feel sleepy. Why? Because in the curd, you are adding in the milk, when you are adding curd, that means you are adding some amount of the bacteria lactobacillus. That lactobacillus get the nutrition alerts there, so they respire there anaerobically and form the ethyl alcohol. So ethyl alcohol there is, and ethyl alcohol that will induce the sleep to you. Okay, so that was the example. So say here solidification of egg and the curdification of milk is the example of denaturation of protein. Okay, guys. Okay, now let's see about this question. Question is, hallosis, why melting point and solubility increases? So what is here? Very simple here. They are bonded with each other through hydrogen bonding. That is where the water goes in the water, in the protein there is in the eggs there is water. So where that water goes when you apply the heat energy. So like amino acids there in the protein, there is NH bonding. So water will act over that and will dissociate a long chain polypeptide into their small chain polypeptide. So hydrolysis of the protein, that means the water present in the eggs, that will be used in the hydrolysis of the protein. As a result, the long chain polypeptide chain will convert into short chain polypeptide chain. So there water will be used. Now let's example here is enzymes. What is enzyme? So enzyme are, one point is here, they are proteinous in nature. Enzymes are made up of protein. One thing always keep in your mind. Okay, enzyme is a protein, made up of protein. That is protein actually, that is, and that increases the biochemical reaction. So enzymes are the chemical substance, that means enzymes, the proteinous substance which enhance the biochemical reaction. That is called enzymes. In the examination, less chances of asking that define the enzymes, but you should keep in your mind that enzymes are the proteinous substance which enhance the biochemical reaction. That is enzymes. And here, example here is like that is maltose here. On the maltose, the enzyme maltase. Okay, one point here. How we write the name of the enzymes? The name of the enzymes are based on 2 points. One is that the substrate on which the enzymes act. During the reactions, we ask them the reactant here, that will call substrate. So enzymes are named on the basis of substrate on which that act, and the suffix ase, age. The suffix is are added in the name of that substrate. And second one is like that when it acts on maltose, that will call maltase. When it will act on protein, so that will call protease. Okay, so if the enzymes will act on the protein and will dissociate them into the polypeptide chain, then the enzyme will be called protease. So same here. And second one is that if the types of reactions in which the enzymes participate, like if the enzymes participate in the reaction of oxidation, then the type of that name of the enzyme will be named as oxidase. In the same way, if the enzyme will cause the inversion, then that will be called invertase. Okay, same when the enzyme will enhance the reduction reaction, then that will be called reductase. If the enzyme will act on the redox reaction, then that will be called oxidoreductase. That is, now example here is about the mechanism of enzyme action. So what is here about the mechanism of enzyme action? Then after, as a particular lock can be opened by a particular key only. In the same way, a particular enzyme will act on a particular substrate only. Why a lock can be opened by a particular key only? Because the active site of lock, suppose this is a lock here, this is a lock, here is the active site. Active site means the portion in which you insert the key. So the shape of your key, the shape of this key, this one active site of key and that of the active site of lock are complementary to each other. Complementary to each other means that this active site of this key can be fitted in the active site of this lock. Okay, so this enzyme could be opened. In the same way, the enzyme and substrate, the active site of the enzymes, only a particular enzyme, the enzyme will act on only those substrate whose shape will be complementary to that of the active site of enzyme. Okay, so that is mechanism of enzyme action. And second one is enzymes are highly efficient. Why? Because enzymes are highly efficient because in an enzyme there is a number of active sites. In an enzyme there are a number of active sites. That's why the enzyme, so a number of substrate would be fitted in that enzyme. As a result, a small amount of enzymes could perform the reactions, could convert a number of substrate in their product. So enzymes are, that's why, highly efficient. Okay, and catalyst, what's about the catalyst you know already. So enzymes are biocatalyst. So obviously, about the catalyst you know. So it increases the rate of reaction. That is here, highly efficient. Why is that? Second one is here, that is about the mechanism of enzyme action. Okay, so that was about the enzymes. In the next class, we will discuss about the vitamins. Thank you, Team Educate.